Hello Pharma Warriors. Today, we're doing a rapid fire session on size separation in pharmaceutics. I'll give you 7 MCQs with explanations, so you can test your knowledge and revise fast. Make sure to pause if you need to think, and let's get started. Which of the following techniques is used for separating coarse particles from fine particles? A. Sedimentation. B. Sieving. C. Centrifugation. D. Filtration. Pause 2 seconds for thinking. Answer: B. Sieving. Explanation: sieving separates particles based on size using mesh or screens. Coarse particles stay on the sieve and fines pass through. Simple, fast, and widely used in powder processing. Which method uses gravitational settling to separate particles in a liquid? A. Centrifugation. B. Sedimentation. C. Filtration. D. Sieving. Answer: B. Sedimentation. Explanation: in sedimentation, heavier particles settle at the bottom due to gravity. It's commonly used in suspensions to remove insoluble solids. Remember, slow process, but simple and cost-effective. For very fine particle separation, which method is most suitable? A. Sieving. B. Filtration. C. Centrifugation. D. Sedimentation. Answer: C. Centrifugation. Explanation: centrifugation uses high-speed rotation to create artificial gravity, separating very fine particles that don't settle easily by gravity, essential for colloids and nano-sized particles. Which of these techniques can separate particles based on both size and shape? A. Sieving. B. Sedimentation. C. Air classification. D. Filtration. Answer: C. Air classification. Explanation: air classifiers blow air through particles, separating them by size and aerodynamic shape. Very useful in pharmaceutical powders to get uniform particle distribution. Which method is preferred for separating insoluble solid from liquid quickly? A. Filtration. B. Sedimentation. C. Centrifugation. D. Sieving. Answer: C. Centrifugation. Explanation: when time is critical and solids are fine, centrifugation speeds up separation compared to sedimentation. Often used in blood plasma separation and lab formulations. What is the principle of sieving? A. Difference in density. B. Particle shape and size. C. Gravitational settling. D. Solubility difference. Answer: B. Particle shape and size. Explanation: sieving depends on particle size relative to mesh openings. Shape can also affect passage. Always remember, smaller particles pass, bigger particles remain. Which method is commonly used for micronization of powders before separation? A. Jet milling. B. Sedimentation. C. Sieving. D. Filtration. Answer: A. Jet milling. Explanation: micronization reduces particle size to micrometer range, making separation, dissolution, and formulation easier. Jet mills use high-velocity air streams for particle collisions. If you enjoyed this session, hit like, subscribe, and comment below which MCQ was tricky for you. Keep practicing and stay sharp for your pharmacy exams.